I'm going to do this two ways, and if you want, you can divide the labor at your table into two different subgroups. Do it, all right? Both groups need a slide, a clean slide, and put some water on the slide. Now, I'm going to put very little water on this slide. You'll see why in a moment. Just the smallest drop I can get away with. And this one I'm going to put a little bit more, like that. This one is going to be a traditional wet mount. So it's actually almost exactly what you do with the ostracods, all right? This one's going to be heat fixed. Heat fixed. I'm going to start with that one there. Take one of these devices. I know you want to say it's a toothpick. You're wrong. What do you think it really is?
Cell picker?
This is biology. This is high-end tech science. That is a human simple squamous epithelial cell sampling device.
Can you repeat it again slowly?
No, I'm not. It's an HSSE SD. All right. Very expensive, high tech. Made of carbon, you know.
Yeah, right. Yeah, expensive. Yeah,
For
right.
sure.
All right. So take your sampling device and sample some cheek cells. All right. It's really simple. Just don't stab yourself. We don't need blood samples today. 7th or 8th week, we actually do look at blood, but not today. All right. This is great.
But isn't it cellulose actually?
Carbon in the cellulose, yes. All right, so get your cheek cells on there. You can't see them, obviously. They're really small. And spread them out. I'm doing the heat fixed one first. So you want less water on this. Spread it out as much as you can, because it's important that this one get dry before you do anything else with it. All right, you want the cells in there nicely spread out and getting dry. All right, and then you can take a little more sample, all right, and put it in the wet mount one. Okay, like that.
Okay.
And then you put this in your biological biohazard container, which is way over there. All right, keep it there for now. Okay, this left-hand one needs to get dry first. All right, this one we can start working with. You can do the way with the ostracod, put the cover slip first, or you can put the stain on right now. A small drop in there, like that. This one you don't stain yet. Okay, mix it up. You can even put some more cells in there. Oops, I didn't mean to do that. Yeah, get rid of those extra drops. Just a little piece, tiny piece. I don't need the whole tree, please. Just a little bit. Catch up those little tiny drops like that. Okay, that's good. Try to get that one dry. This one is pretty much ready to go already, so cover slip. You know how to do this already. Spread it out and drop it. Hopefully no air bubbles in there. If you see dark-edged, light-colored circles, that's a bubble, right? And it's not a cell. All right? How many people thought they were cells? It does happen. Now I do want to de-stain this. If you look close, you can see there's some material in there already that's catching the stain. It's darker than the background. I don't know if you guys can see, but if I tried to look at it right now, the background would be blue and the cells would be darker blue. So it's better to get rid of that lighter blue background. De-staining, yes. Okay, so I put some water on the edge like that. You can see it's diffusing in there. And of course, as you do this, some of the cells are getting washed out as well, but believe me, there will be enough left. You can see the material now.
Yeah, some of it has been wiped.
Yep. And use a little bit of tissue here. Not that much. Just fold it up. Try to get that out of there. pretty simple, honestly. So this is your basic wet mount slide of cells. I could actually destain it more. For what I'm going to do, does it matter that I have stain on the right side here? Why does that not really matter about still having stain over there?
Because you're not going to see those.
It's the field of view, right? You know, the field of view is, 1,000 times smaller than the size of that cover slip, right? So if I mainly look at the cells on the left, then it doesn't really matter what's on the right too much. Just good to get rid of some of that water there, so the cover slip doesn't move too much. That's another thing, especially when you put oil on it. The cover slip can slide around, so try to lower it down by taking some of the water out as best you can. Okay, that's not too bad. So you can see, take a look at that. Okay. You can just pass that around if you want. That's what you should get after staining it with the wet mount. All right, like that. You actually see those things. Now the parts you can see on there are probably chunks of cells. They're more than one cell. They're not really suitable for doing the measurements, as they're overlapping each other too much, right?
So that's why it is visible.
That's why those dark blue spots are visible.
So it may not be just a single cell, it may be just a whole lot of cells.
It's a pile of cells. Yeah, exactly. The part you're going to look at, the cells you're going to look at, you really can't see with naked eye at this point. Right? Okay, so that's fine. All right, so that's there to be observed. If you have to, you can look at that first, but you kind of want this one to dry out. It's very important to dry out completely. If it doesn't dry out completely, the cells won't be nicely fixed on there. So give it enough time to really dry it. Now I am going to speed things up a little bit here using the magic tissue, all right? Because I want you guys to get to work. I'm just kind of looking to where there's still water left.
And so some of the cells may be wiped out.
Definitely. Again, what am I doing? What's this called? Dabbing. If you wipe, you will remove all your cells. If you dab, you will.
This is what you call dabbing.
Dabbing is just touching it to absorb the water. Okay, that's almost dry now. I can actually see, I don't know if you can see, if you look at an angle, you can see there's quite a bit of material. You have to look at the way the light's reflected.
I see.
There's quite a bit of material stuck there.
Yes.
Can you see it? It's a little bit rough on the glass. All right, so the next step, when this is really dry, we're going to heat fix it. All right, and heat fixing is way of cooking the proteins, basically, that are in the cells. And by cooking them and denaturing them, they become solid, and that makes the cell usually more visible under the microscope. When you put the stain on, everything stays in place. All right. You can actually try this with ostracot stuff too, if you feel excited. Cook your ostracot a little bit after squishing it. Okay, that's almost completely dry now. Almost there. I can see the water disappearing.
I can't see the water now.
Okay, that's about right. All right, so heat fixing now. Oh, we need the forceps. That's what I need. Yeah, forceps like this. Find the ones with a kind of flat end, not the pointy end, and you're going to hold that in the in and out of the flame
So
3
we are
times.
using ethanol, not the benzene, right?
No, we're not
So
using
use the
Bunsen
heat.
burners. Too much for us.
So this is what you call the heat, right?
Yeah.
It's
Yeah.
used fire.
Okay. Okay. So you light your little alcohol burner like that. Okay. and you go barbecue your cells. But they are just cells, and if you ever cooked anything on a fire, you know something this small is going to cook really fast, and you don't want to cook it. If you cook it, they're burned, they're charred, and you have a bunch of black spots on your slide. All right, that's really easy to do. You want to just move it in and out, normally 3 times. All right.
Okay, that's
1,
barbecue.
2, 3. All right.
Okay, barbecue
You
done.
can see it got a little steamy there, and turn that off. All right, make sure you cover that before your friend leans over it, especially the ones with the long hair. Okay.
Disaster.
Yeah, and that is now heat fixed. All right, now it doesn't really look any different than it did before, but I know from experience I've denatured the proteins enough, and now I can stain that in the same way. Make sure you put the stain where the material is.
So how could you call this method? Heat something?
Heat fixing.
Heat fixing. Oh.
This is heat fixing
That's
cells.
why you call heat fixing.
Yeah. Leave it on there a bit, okay? Maybe 20, 30 seconds, 40 seconds, all right? It doesn't take very long. The stain is quite concentrated. Okay, that should be good.
So
And
how long should we
then
leave like that?
20 to 30 seconds, 40 seconds
20
is
to
long
30
enough,
seconds.
all right? The longer you leave it, the more the stain will penetrate in there. Of course, if it penetrates too much, then everything becomes dark blue and it's sort of useless. So now you can just absorb it. The cells are all nicely stuck onto the glass, hopefully.
Dabbing again, right?
Dabbing, not wiping. Okay, so get most of that stain off that way. And then add a little destaining liquid here, which is just water.
hydrogen monoxide.
All right, dihydrogen
It's
oxide.
very dangerous.
Very, very. You can drown, you know?
Yeah.
Okay.
I think
Yeah,
it's
so it's basically pretty good. And then you can actually up to 400x you can observe it as it is right now. At 1,000 you've got to wait for the water to really dry off again and then put the oil directly on it. You do not need a cover slip. Once it's heat fixed like this, you don't need cover slips anymore. All right, so oil directly on there, hopefully no water.
So we denature the protein in order to make it stick to the glass.
Stick to the glass, and it solidifies the cell structures so that they are higher contrast. All right.
So that's why it doesn't need any cold glass, right?
Exactly.
So
Yeah.
this is very important.
So it's still a little bit of material. You can see a little bit of water left on there, but okay. Yeah. So that's kind of what you're aiming at.
Yeah, I
So
see
it's still
cells.
drying out a little bit, but that should be good to look at. I'll put it on a microscope, show you what it looks like later. You're free. All right. Go.